This protocol describes the synthesis of a nanoparticle system that is able to silence genes in vivo by delivering siRNAs with high efficiency. By simply exchanging the targeting peptide and the siRNA payload, the fusogenic nanoparticles may be used as a potential therapeutic formulation for diseases that require in vivo gene modulation. We have previously published results using the Fusogenic nano platform against gram-positive bacterial infections by silencing a gene that encodes for inflammation in macrophages to terminate chronic inflammation.
Prior to performing this procedure, assemble a Teflon etch cell containing a silicon wafer in a 3:1 hydrofluoric acid solution. Run an alternating current of a square waveform with a low current density of 50 milliamperes per centimeter squared for 0.6 seconds and a high current density of 400 milliamperes per centimeter squared for 0.36 seconds, repeated for 500 cycles. Once the etching is finished, remove the cell from the circuit and carefully rinse out the hydrofluoric acid solution using a syringe. Then, rinse 3 times with ethanol. To lift off the porous layer from the silicon wafer, first fill the well of the etch cell with 10 milliliters of a 1:29 hydrofluoric acid solution. Then run a constant of 3.7 milliamperes per centimeter squared for 250 seconds. Once the etching is finished, remove the cell from the circuit. The porous silicon layer may have visible ripples indicating detachment from the crystalline silicon wafer. Gently wash out the hydrofluoric acid solution and rinse 3 times each with ethanol and water. Using a